Hey guys, welcome back to Bioinformatics. We're in the middle of our microarray analysis here. We just kind of loaded everything in and got our script set up, and now we're actually going to dive into a bit of the differential gene expression analysis. So without hesitation, let's get into it. Okay, so this is where we left off. We said, we made a little banner, said differential gene expression analysis. I'm going to also say that this is flight versus ground. And so we're going to start by saying that our condition is our targets. Remember, we loaded previously that target file. And so our condition, if we print this, is we have 3 ground and 3 flight experiments. So now we're going to say that the design of our experiment is a model matrix, which is down there, and we're going to say it's by factor of condition. So what we're saying is compare, all this is really saying is compare our conditions. Ground is one group and flight is another group, and that they're factors. So it's factor meaning categorical and not numerical data. So let's say tell our design that our column names of design are flight and ground. So if we look at our attempted column names shown an object with less than 2 dimensions. Column names of design. Did I run design? There we go. I don't think I ran it. There we go. Okay, column names of our design. So if we were to look at our design, see we have flight, and it's dummy coded as 11111 all the way to 6, and then ground is just 111000. So that's so to look at, if it looks at this matrix, it says if you have 2 1s, you're considered flight. If you have a 1 and a 0, you're considered ground. Okay, so now let's do a regression analysis. So we're going to save in a variable our regression analysis. We're going to call it fit, and we're going to run lm_fit. So as you can see in that small text there, it says it fits a linear model for each gene given a series of arrays. So we'll do fit of our expression set, so of our expression data, and we'll use design as the way we're comparing, so to break it up into flight and ground groups. Now we're going to save over fit. We're going to add to that eBayes, which is like a statistical. Let's see what eBayes says here. So eBayes gives a linear model fit from lm_fit, so it builds on top of lm_fit, and it computes t statistics, f statistics, log odds of differential expression. So this is going to tell us if our linear model that we fit in the previous command, if it's significantly differentially expressed. Okay, so let's run this. eBayes with an S. There we go. So we're going to do options. Just tell me what options is. So options allows the user to set and examine a variety of global options, which affects the way R computes and displays its results. So what we're doing this for is we're going to reduce the digits to 2 for our results, and then we're going to say top table, which extracts a table of the top ranked genes from our model. So this is just going to show us the top differentially expressed genes. Our coefficient is going to equal 2. Number is going to be infinite, so however many have all of the differential expressions. We're not going to limit it, so that's what input is for. And then we're going to adjust. And we talked about this previously. We need a multiple test correction. I believe we talked about previously, or maybe it was in one of the 10 attempts I tried to record this video before. But so if you're comparing, so if we look at this this AFI data, we have 22,000 different probes, right? And each one of those we're running a regression on. And so if you accept a p-value of 0.05, what you're essentially doing is you're accepting 5% false errors or false discovery rates, so false positives. And so what that means is of the 22,000, so if we have 22,810 genes, then with a p-value of 0.05, we'd get 1,100 false positives, right? And so what false discovery rate does is it corrects your stringency of your p-value, so it makes it a smaller p-value than 0.05, say 0.0005, based on the number of tests that you're running. So since we're running 22,000 plus tests, we're going to have a smaller p-value to consider it significant. Really important to do this in statistics, or in bioinformatics, and you'll be able to see the difference once we show what's going on here. Okay, so let's just look at the top real quick. So this was top table, and so as you can see here, since I ran infinite, it's like a million of these things. We have our log fold change, so how a log corrected value of how much higher or lower the expression was, right? And so -3 means that it was downregulated, so in space the genes were turned down by a log fold change of 3.5. A positive means it was turned up by 3.5 in space. We have our average expression or t-statistic. So this is where the multiple test correction comes in, right? So our p-value is how significant this is. And so for gene probe 264005, our p-value itself is 1.4 times 10 to the negative 7, so very significant. But we have to be more stringent because we're doing multiple tests, right? And so once we correct the p-value, we get a p-value of 0.002. So we still use the idea of 0.05 here, but just in the sense of a multiple test correction. So if we were to go, we have a lot of significant ones. Eventually we'd get to some that say they're significant, just the normal p-value, but after the test correction is not significant anymore. So those we just, for the sake of good science, we just ignore. So now we're going to create a new section. Let's combine annotations. So we have our annotations and our gene expression and stuff all in multiple data frames right now, right? So we want to kind of put everything together. So I'm going to make a variable called annot for annotation, and it's going to be a data frame. And this is going to be a long code because there's no like tidyverse way to do it really. So genes are going to be sapply contents of ATH11 gene, oops. Oh, is that right? 501, yep. Gene name. Paste collapse equals that. Okay, so what's going on here? So let me make sure I got it right. Yeah. Okay, so what we're doing here is we're making a data frame, and what we're doing is we're making a column called gene, and we're taking all the contents of the ATH, this database, I'm just going to call it this database instead of reading these numbers, the ATH database gene name category, and pasting it into a column, right? So now we're going to do keg equals sapply. So we're just going to kind of run this over again, but we're going to use path, which is the column in the database for the KEGG pathway, which is another annotation type. Collapse equals going to do Go, which is gene ontology, and so this is more kind of a different annotation that tells you the molecular area, molecular characterization of the gene. So does it reside in the nucleus? Does it reside in the cell membrane? Things like that. So more useful information. Okay, we're going to do symbol. I want to capitalize and keep it on the level here. Contents, ATH, and this was called symbol. So this is just going to take out that gene symbol instead of the weird uniform probe names or tear numbers. And then acnum is the accession number. Supply, contents, ATH, blah blah blah, ACC num, paste, collapse equals. Okay, and then we don't need a comma after the last one because that's the last one we're going to run. So hopefully you can pause it here and write all this down. It's a lot of typing, I know, and I hopefully didn't mess up here. So let's run this. Oh, this. Oh, escape out. All right. So problem. This got shot way down here, and I didn't run it. All right. So you should have 2 parentheses at the end. Okay. What's going on? Oh, it's running. Okay. It might take a little bit because it's calling this database, or like linking up with this database to get your annotation. Okay, so now it's done. So if we look at annotation now, we have our row names as our probe names, sorry, our gene, our Go annotation, our symbol, etc. Okay, so now let's do all as our new data frame, and we're going to merge annotate, and top, if we look at top, we have our log fold change, our average expression, our p-value, our adjusted p-value, which we're interested in, and our beta statistic. And top together, and we're going to say by x equals 0. So we're saying merge it by the row name that is in common. So all the row names that are the same will put the right data in the right row, right? Okay. By y equals 0, meaning so it's going to keep all the individual columns that are different, and then all equals true. So it means keep everything. So it might take a second here because, like I said, there's 22,000 times however many columns we have, 12 columns. So now we have our all data frame where we have our row names, and it's called row names here, gene, KEGG, gene ontology, all that stuff before, but we've also added into the same data frame our log fold change, expression, p-value, etc. Okay. So let's do all 2 now. So we want to merge one more data frame with it. We're going to merge all and AFI data by x equals row names, because remember, if you look at this annotation here, we had it as row names. This isn't like a really real column, that's why it's gray on my screen. But now all, they got put into their own column as part of the merge function. So now we're just going to say we're using the actual row names column instead of that first column. And by y equals array element name. So we're doing row names for our first x. So let me, I explained this wrong previously. So for the merge here, what this is doing is by x means x for annotate, use the column 0, and for top, use column 0 as the way you merge. So when we do it this time, we say for all, use the row names column. So where's all? Row names column. And then for data, use the array element name. And so those should be the same names and annotation. It'll always keep all the columns. Okay, so let's run all 2. So now we have 20 variables: row names, gene keg, gene ontology. We kept all the expression stuff, but we also added some of that other stuff that we didn't have before, right? So the start and stop base pair for the gene, the chromosome, the description, etc. So we've got a ton of really good compiled data there. One thing we had to do quick though, all 2 ACC num, we have to convert this to a character, because when we put it into there, it got kind of coerced into, I don't know if it's a number or a factor or something, but I had to do this to get it to run correctly. So let's make sure that the accession number is a character. And so now what we're going to do is write a table, and you should be annotating as we go here, and I'm of course skipping it. Let's convert the ACC num to a character value. Let's merge all the data into one data frame. Okay, so now let's save our data. So we're going to write a table called all2, and our file is going to be called brick16 final.csv. Row names is true. Column names is true. So we're going to keep the row names and the column names. And our separator, we'll make it a tab. Select the file. You could do a CSV, but I like tabs. And so let's run that. And so it writes that. So if you open up your, where am I working this out of? What did I call this file? Brick16 final. Brick16 final. So it looks like a mess because we're because it's in Excel. What you could do, so you could spread these out, kind of see what's going on. But so we've saved all of our data into one. We backed it up. So if you ever you don't have to rerun the session if you're going to keep running analysis on this, or if you need to send this data to someone else, and they don't have R or whatever. So we backed up all the data, which I would encourage you to do intermittently in your projects. Okay, so let's do one last thing here. So now let's look at columns org.at.tear.db. So as you can see here, for our in this org tear database thing, we have a bunch of different more annotation to our genes, and this is going to be important for the last part of this, where we do what's called Pathview. So we're going to look at the biological relevance of this. And so full changes we're going to create a variable called fold changes, and we're going to say as data frame all to log FC. So where is it? Fold changes. Fold changes. So you can see this is just the fold changes, how high or low it was, right? And so we're going to do all to entres equals map IDs org.at.tear.db, comma, next line, keys equals alt to ACC num, column equals entres ID, key type equals tear, and multi-vals equals first. Okay, so what are we doing here? So we're adding a column to our all2 data frame called entres. And we're using this function called map IDs to look into the Arabidopsis database and find the ACC, or use the ACC num to match up to the entres ID. Key type is tear, meaning that this ACC num column is a tear annotation, so it's got to know what kind of annotation it's using. And then it says if there's multiple values, use the first one. And so now head all to 10. Let's look at what we got here. And so let me pull this up. So we got a lot of data here, and it prints it in multiple rows because, so it's just the first 10 lines, but you can see we have symbol, ACC num, log, fold change, average expression, our statistics, our organism, this locus. We have our address. It should be on here somewhere. How about I just pull it up this way? This is the nice thing about RStudio is it makes it easier to read. So we should have entres. Do we not? Let's see. All to entres. Maybe I did not run it. Did I not run it? Oh, okay. So now let's look at it. All two. Entres. So it says NA for a lot of them, because a lot of them are like hypothetical proteins and things like that. So if you scroll down far enough, you can see the entres numbers. Okay. So the last thing we're going to do is Pathview. And that's going to put some of this into biological context. However, we're going to stop right there. So before we got all of our data loaded in the last video, this video we ran our differential expression and took all of our annotation data and coupled it with our differential expression data. And now what we're going to do after this in the next video, I suppose, is put it into biological context. What is significantly different between space and ground in terms of gene expressions in plants for microarray data. So that being said, I hope you enjoyed the video. I hope everything ran well for you and you didn't make many typos.